team today is going to be focusing on glycolysis, and we'll be focusing on aerobic glycolysis. This is dominant in the fed state when glucose is abundant and is the first metabolic pathway of cellular respiration to produce energy in the form of ATP. It is a 10-step process occurring in the cytoplasm of nearly all cells, and through a series of enzymatic reactions, the 6-carbon ring of glucose is cleaved into 2 3-carbon sugars of pyruvate and 8 ATP molecules. Pyruvate can then go on to enter the citric acid cycle and oxidative phosphorylation in the mitochondria. Through the investment of 1 ATP molecule, glucose is phosphorylated into glucose 6-phosphate, also using glucokinase and hexokinase as catalysts. This step is irreversible and traps glucose inside the cell, since phosphorylated glucose can't easily cross the membrane, which leads to a reduction in intracellular glucose concentrations, which further increases glucose uptake. Glucose 6-phosphate is isomerized to fructose 6-phosphate by phosphohexose isomerase, which means it's just changing the shape from the traditional glucose shape to a traditional fructose shape. And with the investment of another ATP molecule, fructose 6-phosphate is phosphorylated into fructose 1,6-bisphosphate using the enzyme phosphofructokinase as a catalyst. This step is irreversible, and phosphofructokinase is the regulatory enzyme in glycolysis, which can either slow down or speed up the process and determines whether glycolysis needs to continue. An abundance of ATP inhibits the activity, and abundance of ADP catalyzes the activity. Thank you.
The step of glycolysis uses an aldolase enzyme to split the 6-carbon fructose 1,6-bisphosphate into 2 trioses, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Step 5 is the interconversion of these trioses. Glyceraldehyde 3-phosphate is the molecule that continues through the glycolytic pathway. The interconversion happens to maintain equilibrium between the 2 molecules. As glyceraldehyde 3-phosphate is depleted, the enzyme works to help this flow and create more glyceraldehyde 3-phosphate from the dihydroxyacetone phosphate molecule. Step 6 is the beginning of the second half of glycolysis, where there are energy-releasing steps leading to the production of ATP and energy-carrying molecules. In this step, we use the enzyme glyceraldehyde 3-phosphate dehydrogenase. A dehydrogenase works to remove electrons and hydrogen from its substrate. NAD+ is reduced and accepts the electrons and hydrogen in this reaction to form NADH. This is the energy-carrying molecule that will eventually be used in a pathway outside of glycolysis to harvest more energy. So in this step, the starting molecule, glyceraldehyde 3-phosphate, is oxidized at the aldehyde group. NAD+ is reduced to NADH, and then an inorganic phosphate is added. The molecule formed at the end is 1,3-bisphosphoglycerate. This is really important because the phosphorylated aldehyde group is a high energy group called an acyl phosphate. This is the molecule that continues on into the next step of glycolysis.
Step 7 of glycolysis is going to involve 1,2-3-biphosphoglycerate and converting that to 3-phosphoglycerate. So this is going to involve the phosphoglycerate kinase enzyme. This is a substrate level phosphorylation as the phosphate is donated by the substrate, and an example of a reaction where ATP can be produced without involving the electron transport chain. So this is going to be the first ATP that is going to be formed, and as glyceraldehyde 3-phosphate is going to be ran twice, we're going to have 2 ATP from this step. Then we have step 8, which is going to be the conversion of 3-phosphoglycerate to 2-phosphoglycerate, which is basically just a shift in the bond position, and the third position is going to become the second position, and the enzyme is going to be mutase phosphoglycerate, which is going to be a readily reversible reaction. Then we have step 9, which is going to be the dehydration of 2-phosphoglycerate, and it's going to be converted to phosphoenolpyruvate with the release of the water molecule, and it's going to involve the enzyme enolase, which is going to require Mg2+, as phosphoenolpyruvate is going to be formed. This is going to be a high energy phosphate bond. Then we have step 10, which is going to involve the conversion of phosphoenolpyruvate, which is going to remove the phosphate, and it's going to allow for the formation of ATP by the enzyme pyruvate kinase. This is going to be a regulatory enzyme that is involved in glycolysis, and it's going to be activated by ADP and AMP. And this step of dephosphorylation of pyruvate is going to be irreversible. So now we have ADP becomes ATP, and this is going to be the second substrate level phosphorylation, which is going to form 2 ATP molecules. And now we have a net formation of 2 pyruvate molecules. Sources and references listed here. And we want to thank you for watching our presentation of glycolysis in our metabolic pathway. Thank you.